I'll open up yours. No, that's a problem. Good. Nice. Take it away.
Okay, hi everybody. Like Gord said, my name is Kerri Lynch. I'm working under Matt Robertson, and today I'm going to be talking to you all about my project, which is a new diet-based index of abundance for 2J3KL capelin. So today I'm just going to go through a pretty quick introduction, what some of my research questions are, and I'm going to spend most of my time kind of discussing the project, how the project's been going, and the logical steps that I've kind of gone through as I've developed this project. And then I will just briefly wrap up with the next steps and where I want to go from here. So to start off, I'm sure we're pretty all aware of what capelin is, especially with the capelin stock assessment just happening last week. But to get it fresh in everyone's mind, capelin is a small forage fish species that has circumpolar distribution around the Arctic. So it's found in the North Pacific, in areas like the Japanese Sea and Western Canada and Alaska. And it's also found in the North Atlantic, including the Barents Sea and Norwegian seas. And also, importantly, it's found around Newfoundland and Labrador. And this is important for me because I'm going to be studying the NAFO division 2J3KL capelin stock. And this stock kind of is in the region of the northeastern Newfoundland shelf and the northern Grand Banks. So capelin is an important fish for the fishing industry here in Newfoundland, with peak catches being around 250,000 in the late 1970s. But it's also a very ecologically important species. The Newfoundland and Labrador marine ecosystem is often described as having this wasp-waste structure, where capelin is this intermediate species that moves energy up the food web from primary and secondary producers up to large vertebrates like Atlantic cod, marine mammals, and seabirds. Unfortunately for this 2J3KL capelin stock, their population collapsed around 1991 with limited recovery since. And the cause of this collapse and subsequent lack of recovery remain poorly understood. Like most stocks in Canada, or all stocks in Canada, DFO does stock assessments every year or every few years, depending on the stock that we're looking at. But for this stock in particular, they do an acoustic survey in the spring in 3KL, and they have a forecast model of abundance that uses larvae from Bellevue Beach in Trinity Bay. And they also have this fall survey, trawl survey that's done in the fall that goes through all of 2J and 3KL. But it's not really used for abundance. It's more used to make sure that the trends that we're getting from our acoustic survey and our forecast model all agree with each other. Why are we looking at making a new model for capelin abundance if we already have these 2 or 3 other models that work pretty well? Well, for one, the acoustic survey is only done in the spring, so we're kind of missing a lot of the temporal variation that could happen with the capelin population. And also, most of the capelin population exists in this like northern Grand Banks area, which is the 3KL area, but we could still be missing a part of the population. And then the other question that I kind of come up with is, what if just the other models are wrong? What if we're missing data some years? What if it just doesn't work out? And we can see here in the graph on the right that there are some years where we just don't have estimates because something has gone on with the data. And so what I want to do is establish a new index of capelin abundance post-collapse using predator stomach contents. To do this, I need to compare a diet-based index of abundance to an acoustic-based index of abundance, which is what we're currently using. To make this diet-based index of abundance, we need to determine, are there any factors modifying the functional response of predator-prey relationships, because this functional response is what we're going to base this diet-based index on. To make this functional response, though, we need to be able to relate predator stomach contents and bottom trawl data to each other. And this last thing is what I'm going to focus most of my talk on today. I just want to take a second and let everyone breathe because I will be talking about math a little bit, but it should be pretty simple and I'm sure we can get through it together. So just to start off with, when we're looking at predator-prey relationships or predator consumption and prey abundance, our classic approach is to look at it as a linear relationship. And so we're saying for all of like the number of prey that we find in some predator's stomach, that is equal to the amount of prey that is in the environment. However, this model kind of assumes three things. It assumes that there's unbiased counts. It assumes that this linear relationship is in fact a linear relationship. And it also, because it's such a simple model, it assumes that there's no outside effects that could change this relationship. I'm going to go into each one of them, and we're going to start off with this unbiased counts assumption. So my issue with unbiased counts is that there could just be errors in the counts themselves, whether that is caused by digestion. So if prey is in a predator's stomach for too long, it could partially or fully digest, and we wouldn't be able to identify that as an actual positive prey in a stomach. The same thing with stomach evacuation. When predators are brought up from the bottom, their stomachs can actually just eject prey, and so we wouldn't be able to get correct abundance estimates. So to address this, we're basically going to change it from counts to binomial presence-absence data. So whether there is a prey in the stomach or not. Let's move on to whether this linear relationship is actually a linear relationship. Linear relationships kind of assume that the predator is consuming every individual of that prey species that they encounter, which we know probably isn't true, most likely not true, if we think about alternate prey being in the environment. So whether there's a predator and two or more prey species, that predator is not going to consume every single prey it encounters. It might have a preference for one species or another, just depending on different factors. And so in that case, we're going to use something that's called a functional response. And this was originally defined by C.S. Hulling, and it describes the relationship of a predator's consumption of prey at different prey densities. Generally, it's classified into three categories. The first is this linear relationship that we were talking about before. The second is an asymptotic relationship where predators are limited by the ability to process food. And the third is this sigmoidal shape that we see here. So now that we've changed from using this linear relationship to a functional response, we need to make a new equation that utilizes predator functional response instead. And this is the equation here. So R is the rate of consumption of the prey. I is a shape parameter. Beta describes the form of the curve. So that's whether it's type 1, 2, or 3, like we were just looking at. And then D is the density of prey, which is described by the trawl, which is what I was mentioning before in the introduction. And the same thing for trawl data, because of unbiased counts maybe, we're going to reduce this to binomial presence-absence data as well. We've now addressed those two things. Let's move on to the third of the assumption that there are no outside effects. Now that we're using predator-prey relationships to estimate abundance, we're assuming that there's no outside effects changing the predator's functional response. And when we think about the functional response and when we think about what could change a predator's consumption rate, I'm thinking things like temperature. I'm thinking that different lengths of a species can influence how much they're eating. And I'm thinking that different species might consume prey differently. And when we actually look at these functional responses that we get when we add temperature into a model, where the black line is a model that doesn't include temperature as a factor, compared to the red line where we do include temperature as a factor, there is a big difference between the shape and also the magnitude of consumption rates between them. With species, we don't see as much of a difference in the functional response. Could be due to the data that we have right now. But there still is a difference, so it's interesting to look at. And then the last thing, when we're looking at length, we see that these top two length bins, which is the brown and the green, they actually consume much more capelin at higher prey densities than the smaller length bins that are the ones on the bottom. Okay, so we've kind of addressed all of these different assumptions. We've changed our biased counts, potentially, or unbiased counts to binomial data, we're now using a functional response and we have to add covariates. And so now that we've changed from this like linear model to a nonlinear functional response model, what's next? What's next is we have to figure out a way to integrate all of these different covariates into a functional response to create an abundance index. What's this abundance index even going to look like, though? And we can see that this is what we're going to basically find out, where the photo or the graph on the left is the model or the abundance of capelin over time without any covariates added. And so you can see that there's a lot of interannual variation, but it relatively stays the same. Whereas when we look at the right graph where temperature is added into the abundance, there's a lot less interannual variation, and the trends between the two are mostly the same, but they do change. And so if we add in all these different covariates, what is this abundance index actually going to look like? And hopefully it'll give us a more ecologically based abundance index of capelin over time. But I'm not sure what it's going to look like. And hopefully by my next presentation, I will know what it looks like and I'll be able to show you all. But for now, I just want to say thank you.
Any questions?
So if your method is able to eliminate biases in terms of what's in the stomach, biases so a small grain of control, why not take it back in time to earlier control, right? Where the assumption is that gear is so different that it might have to catch a small or large species. But if you have those biases removed, what's stopping you from the pre-collapse?
Technically nothing, but we're still in the process of like fully rounding out how to even add in covariates into the initial functional response model. And so I think like once we can add in environmental stuff, we can kind of project backwards and see if that also holds true if we use a different type of trawl data, because right now we're only using data from the Capelin trawls, which starts in 1995, like you were saying. And so hopefully once we've kind of established that it works for this type of trawl, we can see if it works for multiple different types of trawls.
Thanks, I enjoyed both presentations.
So you're switching your density to binary data, but would you still be kind of concerned about like false negatives or false positives, but more so false negatives, where it's like the prey could have been digested and is actually present, but you still can't tell?
Yeah, so that's kind of a tricky situation in that if it's not there and it's not noticeable, then obviously we can't look at it, but we're trying to kind of restrict the data in that we're focusing only on trawls where, or like areas where capelin is known to be present generally. So whether it's at like great depths, capelin only go to around like 400, 500 meters. And so any trawl depths below that, we're not going to include those into this because it will mess up how the model is calculating kind of the functional response curve. And so we're not necessarily like including only predators that have an overlap with capelin, but we're just trying to see if there are capelin in the area and predators are in the area, then hopefully they'll overlap. But yeah, we can't basically say like, you know, that fish is a capelin, but we don't know what it looks like kind of thing. So yeah.
Right on. Thank you. Great presentations, Bob.
Thank you.
All right. Did you send me an email?
Kotliarov? You can say Antonia and then pause and I'll say my last name. Do you want to do that?
Oh, don't drive.
Oh yeah. Yeah.
Questions? Okay. That's all good. All right, great. So next we have Antonia Kotliarov.
Kotliarov.
Okay, thank you. Sorry. who is a first-year fisheries science and technology master's student. She's a part of MI's Florida Ocean Lab and Dal's SEAM Lab, and she's co-supervised by Kathleen Roberts and Craig Brown, and is studying the effects of submarine underwater discharge on benthic communities off the coast of PEI. She completed her bachelor's in marine science at the University of Maine, where she learned that disturbing stuff on the seafloor to get a better look at it is the best use of her time. So with that, I will. Good to go.
And is it just like I click?
Yeah, should be.
Sweet. Hi, I'm Antonia, and I'll be talking about the effects of submarine groundwater discharge on coastal benthic communities of PI, which is my project. So let's start off with what's submarine groundwater discharge? Well, it has absolutely nothing to do with submarines. Very sad. Instead, submarine groundwater discharge, SGD for short, is scientifically defined as any and all flow of water on continental margins from the seabed through the coastal ocean, regardless of fluid composition or driving force. So basically, it's when water that is fresher than seawater flows out from the seafloor and into the surrounding coastal environment. So how that works is you'll get water from rain or other forms of precipitation hitting land. That water then trickles down through the sediment and gets held in something that is called an aquifer. Aquifers are structures made out of porous rocks and soil, which hold and transport groundwater. These aquifers can extend to the coast and far offshore, which allows groundwater, which is this rainwater that just trickled down below ground, to seep out in coastal environments. And here you get areas of submarine groundwater discharge. So we have rainwater that seeps down, turns into groundwater, seeps out as groundwater discharge. Why do we care about all this? Here's 3 reasons why maybe we should care. Groundwater discharge acts as a link between terrestrial and marine environments. So groundwater has a different pH salinity and is oftentimes cooler than the environment it seeps into. It also transports a lot of land-based materials to the seafloor. So groundwater can contain high concentrations of nutrients needed for primary productivity, like nitrates and phosphates. It can also carry dissolved gases, metals, and land-based derived pollutants. And all of this fun stuff seeps right out across the seabed, which leads us to our second point. It plays a significant role in influencing the benthic ecosystem. All the new conditions carried by the groundwater interact with the bottom community first before making its way up through the rest of the water column, which makes benthic communities vulnerable and susceptible to any changes in their environment caused by groundwater. So differences in pH and salinity can influence geochemistry and mixing along the bottom, and can also create estuary-like conditions at the sea floor. And some organisms just can't tolerate that, which can lead to shifts in community dynamics, which might either be helpful or harmful to these coastal ecosystems. Which leads me to my final point, which is that the effects of submarine groundwater discharge can either be good for the ecosystem or they could be bad. It really depends on the physical, geochemical, and biological makeup of the seabed, as well as the strength of groundwater discharge. So studies in like Japan have found that groundwater discharge increases primary productivity and helps a lot with local aquaculture. Some other studies somewhere else in the world, I can't remember where, have found dead zones linked to this groundwater discharge, as well as a decrease in benthic diversity and coral bleaching. So you really don't know what you're going to get with this phenomenon. So my study will focus on discharge found around 16 kilometers offshore northern PI, right here. What we know about this area is it's a big fishing zone. Specifically, it's in the southern Gulf of St. Lawrence. It's the big fishing zone for lobsters, LFA 26, which live on the bottom near the coast. So they could be kind of influenced by this discharge. And one other thing that we know about this study site is we don't really have a lot of data on the benthic community there. As of right now, from what I could find, that could change if I read a different paper. But, so what we're going to try to answer with this project is how submarine groundwater discharge, if it does have an impact on the benthic community, and how it impacts its structure and function, as well as any impacts it may have on the ecosystem services provided by the local benthic community of northern PEI. Knowing whether or not freshwater discharge is enough to influence the benthic community is important in understanding the role it plays in PEI's coastal ecosystem, and it should be considered, and if it should be considered in future management decisions. Compared to other studies on groundwater discharge, this one will focus on its impacts on a community level rather than on an individual species level, which is what has been the focus of other studies in this field. And we're just hoping to add more information to this growing field of groundwater discharge and its impacts on the global marine systems. Cool. So kind of background of what's going to happen. So what already happened is fall 2021, an Ocean Frontier Institute led project called SOURCE, which is studying groundwater around PEI, found seepage locations, which is where my project's going to be centered around. And they have some initial bathymetric data for this site. So currently I'm pinpointing those locations and creating kind of plots of where we're going to go this summer, and then do some CTV tests to see how much fresh water is in the water column, get some more bathymetry data to get a finer scale image of the bottom of the seafloor, and then figure out what's actually down there, on who's living down there, and then combine that biological data and geomorphic data to create a benthic habitat map, which will hopefully help us understand how the benthic community changes around this groundwater discharge site. Yep. That's it.
Questions?
Yes.
So you can show me the schematic. This kind of look like almost like a point source. Your map as well, like a sort of a point source. How big of an area might this sort of site cover? This discharge.
Not that big.
That it?
Yeah, I can't give you an exact number, but it's like it doesn't look like it's huge.
To be sort of straightforward then to look at sort of site versus different site outside the site, that it's not something for a huge.
Yeah. So I'm hoping like point source, and then look kind of like coastline, and then look kind of parallel, and then maybe like perpendicular to like see how everything changes like closer and like with a distance away from this point source. So
Nice. Sounds good.
I was wondering how much of an effect do you think the like rock type, the aquifers can have? It seems kind of new, so I don't know.
like well,
The water is coming through.
great question. I have no idea, but if the rock is more porous, then like it'll probably allow for greater amount of groundwater to discharge. We're not really studying that, so like we truly don't know how much water we're gonna find. Maybe there like won't be any, or like won't be enough to make a big impact. So don't.
Right now. Perfect. Thank you, Antonia, Hannah, and Kerri, and everybody else who joined us. If you have any further questions, I encourage you to reach out to the respective students when you have time. Our next seminar is going to be next Wednesday, March 20th at 12 p.m. featuring Dr. Peter Kickert. We'll live stream in this room and we'll be live stream on WebEx for those who cannot make it in person. So thank you so much, and you can find this recording on our YouTube channel. Perfect. Thank you, everybody.